All right, welcome in on the YouTube side. Let's go ahead and restart the TikTok side as well. And we should be up and streaming on both sides. Thank you guys if you are jumping in for the first time. My name is Matt, and I am one of the most experienced MCAT tutors in the country. And today we are going to be dropping a new kind of live stream. We are trying out. We're going to go through some of the recent study packs that we have been dropping for free on the high yield notes for the MCAT. And what I'm going to try to do on both the YouTube and TikTok side here is we're basically going to put up on screen some of the segments from, see if we can get this to crop down and display, right? Some of the segments from the study packs and just go through them. Hopefully the screen size and display size is convenient for everybody. I am not entirely sure if it will display the same on the YouTube side, but I think I have it sized so that it is somewhat comparable for both the YouTube and TikTok side. If you guys have a question, do be sure, if you have an MCAT or pre-med med school app question, we will pause for questions that get asked in the chat. Also, drop a contact form for personal MCAT. All right. For MCAT help. All right. Okay. We'll jump right back into here. It looks like everything is there. Let me copy this comment over for just 1 second. Pin comment. That way everybody can see that. Drop your questions here. .mcatboss.com for help on the MCAT. All right. Here we go. Okay. So back to our MCAT class. So let's chat about gram stain. This is something that the laboratory techniques are something that students tend to either way overstudy or completely ignore. And I want to encourage you guys to not do either. Like, don't overstudy the lab techniques and don't ignore them, because there are a percentage of questions on every single MCAT that are about lab techniques. Sometimes the questions are direct, like, what are we staining for? And they're usually not that direct, though. They're usually some kind of question dealing with the setup of an experiment where the difference between the correct and incorrect answer depends on you noticing that there is a mistake in the way that they describe an experimental setup, or where you have to just know what the outcome would be based on that lab technique. And there's a couple that are specific to the cell type that we're discussing here. There are a couple that are specific to different types of chemistry experiments. And when they pop up, give them a few minutes, learn them the first time, and then usually they're not too hard to navigate on the actual questions, but they're very, very popular. All right. So from our quick notes here, the gram-positive purple stain. This is the famous one from all of the bacteria, the high-yield bacteria stock photos. Somebody on the YouTube side says, which subject is this? Tonight's MCAT class is about cell biology on the MCAT. Great question. All right. Okay. Very cool. Very cool. Okay. You guys, thank you for jumping in. I see a bunch of people jumping in on the TikTok side. Thank you so much. Drop a like. Let's get this shared out. Dr. Austin says, never the MCAT, never again. Yes. I get the sentiment. Lots of people feel that way once they get through it. Never looking back. All right. The gram-positive purple stain, of course, is referring to how the peptidoglycan wall looks this sort of very intense dark color, sort of a violet iodine look after we decolorize it. The main thing here is that because there is not an outer membrane, the outside of the stain stays purple. And so gram-positive bacteria lack that outer membrane. This is super important to remember because it's a little bit counterintuitive. If you imagine that they're staining for the outside cell membrane, it's a little bit counterintuitive, but they're not staining the outside membrane. They're staining the cell wall. That's super important. Now, a few examples include staph, strep, bacillus, clostridium, and listeria. Some of the 5 biggest heavy hitters in the gram-positive bacterial world. The cell walls of these bacteria, of course, are targeted by beta-lactam antibiotics like penicillin and cephalosporins and vancomycin. So this is a huge part of why the penicillin antibiotics are so incredibly popular, because if you ever hear about somebody having a staph infection or a strep infection or even a listeria infection, the reason that the penicillin and cephalosporin antibiotics are so popular is that they are attacking that cell wall. So the spore formers like Bacillus and Clostridium, of course, are able to form protective spores, and this is a way that they survive heat. They are able to survive a really shocking amount of UV radiation and even desiccation. Drying them out and exposing them to, alcohol also does not seem to take them out. And let me close the live center thing here. Awesome. Okay, so the next segment here is, and let's go this way on our little info sheet. Let me just update the, let me get to the right screen on the YouTube site as well so everybody can follow along on both platforms. And our next segment here is on, I gotta figure out how to crop this on the YouTube side. Okay, our next segment here is on gram-negative. And of course, the gram-negative ones, when you have both gram-positive and gram-negative bacteria, you'll see that deep purple color on the gram-positive ones. And on the gram-negative ones, you will see sort of a pink-red color. And so this, of course, is because they have a thin peptidoglycan layer, which is that hard cell wall, followed by an outer membrane that has the lipopolysaccharides. And of course, these are famous for having the ability to be an endotoxin in humans who are infected with these. The violet is mostly washed out because of that outside membrane. And so the reason that you get that pink and red layer is not actually because that's the natural color. It's really easy to see a picture like that and think that that is the natural color, and that is not the natural color. Instead, the safranin is it's basically a counter stain. It's if you did not pick up the purple stain in the actual cell wall, you go ahead and add this in as a way of demonstrating that you have gram-negative. The reason for that is cells are surprisingly clear. So if you look at a microscope slide with something that's not stained at all, it is really difficult to notice anything because cells stain clear. And so gram-positive pick up purple, and then the safranin basically stains the remaining ones red. Thank you guys for hitting the likes. I appreciate it. Let's get this out to a few more people. All right. Famous gram-negatives include E. coli, Salmonella, Neisseria, Pseudomonas, H. pylori, and of course Helicobacter, the famous little buggers that like to live inside of our stomachs. The reason that we are mentioning here about the lipopolysaccharide trigger is this is something that is famous. This is going to stick inside your mind. Thank you guys for hitting likes. This is something that's going to stick inside your minds forever and ever and ever. Lipopolysaccharides drive septic shock. Lipopolysaccharides catch onto these little hook-shaped proteins inside your intestines. They're called toll-like receptors, so TLRs, and they cause a cytokine storm. A cytokine storm is basically a signaling molecule that blooms inside of your blood if you have one of these septic shock driving lipopolysaccharides, which are the endotoxin created by these LPS containing bacteria. And of course, septic shock is an ongoing problem that happens every single day, dozens of times a day, in lots and lots of people at every major hospital in the world. And Keen says that he prefers his bacteria in his Greek yogurt. Yes, those are the friendly ones, thankfully. All right, the problem that we deal with, and the reason, not just because of septic shock, the reason why these gram gram-negative, these anti-staining bacteria are problematic is because the cell membrane on the outside means that their cell wall doesn't burst when you give them a lot of the classical antibiotics that target peptidoglycan. So things like penicillins don't work on these boogers, and that is a problem because this gives them just a intrinsic resistance to a lot of antibiotics. This means they don't even have to have acquired resistance. They don't need the fancy cell pumps that pump out the antibiotic from inside the cell because they just don't get the antibiotic inside of them at all. They're protected. They don't rupture the cell wall because they already have a way of defending against it. All right. As our gram positive and gram negative, jumping on, let me crop this down a bit so we can see where we are. And let's get to our next topic here. And pardon the blank screen for 1 second, guys. We will get to bacterial gene transfer in 1 second. Let me just update the screen on both the YouTube and TikTok side so everybody can follow along. And jump over here on this side as well. There we go. Perfect. Okay, I think I finally have the display figured out on the YouTube side. I promise you guys this will get smoother as I figure this system out. All right, bacterial gene transfer is our next high yield area. And again, if you guys are following along on the TikTok side, I'm so sorry. I'm going to restart this one more time. Thank you for helping get over 50 likes. We're hopefully everybody is following along, but just the live will go off for a second. If you can jump over to the profile for 1 second, we were going to restart this because it again is having us restart this. All right, 1 second. See you guys in a minute. If you are on the YouTube side, it should keep going. It looks like everything is chugging along perfect on the YouTube side. All right. So we are back up, and on the TikTok side, I see you guys jumping back on immediately. Thank you so much. Sorry for having to restart that. If anybody in the audience happens to know why TikTok, why the TikTok algorithm for some reason, I don't know if it is my face or my setup or my voice or something else. No matter what I have on screen, I have taken absolutely everything off the screen except for the text right over the top of my head that says MCAT Class. And for some reason that I cannot figure out, it keeps popping up after exactly 6 minutes. Every single time it is exactly on the 6 minute mark, it pops up some kind of message saying that the system thinks that I'm reusing content or something. I don't know what that is about, but if anybody knows, please drop a comment. Let me know. Getting back to our quick study guide here. These, again, if you have not gotten these are just being posted on the. These are just being posted on just the main profile. I'm figuring out how to kind of cross post them. If you guys are following along from the YouTube side, I'm going to try to post these as a series of like community posts on YouTube. I think there's 200 or 300 of you guys who follow along on YouTube, and we'll try to get these over to the YouTube side as well. If you're on the TikTok side, if you can see the posts, you will see periodically these posts that have kind of in the range of like 10 to 15 slides, and they're just these hopefully helpful bullets on what the high yield topics are for that particular section, and hopefully they're helpful as you are going through your MCAT class or going through your individual prep, whatever method you are using. All right, so bacterial gene transfer. I wonder if there's a way. Is there a way to write on screen? I don't know if there is. That would be cool. I'll have to figure that out later. All right. Okay. So bacterial gene transfer is really important when you're studying how bacteria manage to survive a lot of the things that we do to try to get rid of them. Bacteria evolve really quickly, and they do it through a number of different mechanisms. One way that bacteria evolve really quickly is that they have a very high mutation tolerance. in humans, we have a relatively strict DNA polymerase enzyme that is pretty good. It's not perfect. Everybody still inherits maybe somewhere in the order of around 100 mutations that their parents did not have. And when a bacteria copies, instead of it only having like 100 mutations out of a 2 or 3 billion base pair genome, bacteria actually naturally, their baseline for mutations is really a lot higher than in humans. But besides that, they also have ways of getting genetics from their neighbors. It is a little bit like you may have read that some humans have the genetic ability to have more oxygen carrying capacity because of the unique genetics behind their hemoglobin, if they come from a group of humans who lived in the very high altitudes, like people whose ancestors lived in the Himalayas, or people whose ancestors lived very high in the Andes Mountains, often have selection for gene variants that give them higher oxygen carrying capacity. Now, if you can imagine hiking up to Machu Picchu and the person next to you with Peruvian ancestry just being able to get more oxygen out of the air, if you can imagine that you could simply absorb some of their genome and have that capability, then you probably understand pretty well the world that bacteria live in, because bacteria can actually do that. If bacteria are in a petri dish or in a person and they are exposed to an antibiotic and one of the bacteria has resistance and one of the bacteria does not have resistance, there are a couple ways that the future generation of bacteria can end up with the genetic resistance to the antibiotic. And one way is the one who already has resistance will just go ahead and make lots of copies of itself. And so the descendants of that bacteria who has that extra plasmid of DNA giving it resistance will have like 10 billion offspring, and those 10 billion offspring will also have resistance to the antibiotic, which is not good for us as humans, but is great for the bacteria. If you guys tap that like, it will get this out to more people. And be sure, if you are interested in going to med school, ask your MCAT and pre-med questions here, and we will pause class to answer them. And of course, also check out MCAT Boss and drop me a line if you need help on the MCAT. And the other thing, though, is besides just getting the descendants of the bacteria who already has resistance, you also have the ability for the bacteria that has the resistant plasmid to give that genetic material to other organisms. And so this happens through, most famously, conjugation, which you see down here in the third bullet point here. That's direct cell-to-cell contact. And again, right on the money at 6 minutes, they have decided to tell us that this is all right, there we go. All right, okay. Now, the conjugation where the bacteria actually connect is one of the ways that they do this. The other way, of course, is that you can have bacteriophage viruses, which interestingly, bacteriophage viruses are really interesting because bacteriophage viruses appear to be trying to hurt the bacteria, but in their attempts to splice their way into the genome of the host bacteria, then when they end up trying to get out of the genome to make offspring viruses, they actually carry some of the genetic material with them, and they can then accidentally give the genetics from their previous host into their next victim. It's really interesting. Genetic transduction is super, super, super interesting. And of course, the other thing that they can do, and this is something that I find, in my opinion, this is probably the most, I think is the most interesting one, is that they can do transformation. And that means if you simply have the DNA swimming in the petri dish next to one bacteria, they can go ahead and take it up and use it for themselves. Okay. we are going to end the live here again because they keep on popping this up in live, and we will keep the YouTube side going. And then what we are going to do is we are going to restart the live here, and I am going to see if we can get it rolling on here. All right, let's find out. All right, guys, if you're following along on the YouTube side, we are going to keep things hopping on the YouTube side. We're going to keep going through the notes here. What we are going to do is because the live keeps crashing in the actual live streaming software through TikTok, we are going to let the live keep on thriving on on the YouTube side. And instead, we are going to finish the TikTok side of the live from the mobile device. And my apologies, guys, on just the absolute craziness of TikTok Live Studio crashing from the desktop. Hopefully, everybody's able to rejoin us here. Also, while we are waiting for people to find us here, we'll take a second and... let's see, we will take a second, and there we go. We're going to announce this on the live. Hopefully that will help more people to find us. And while we get everybody back here, I think what we're going to try to do is, I'm not really sure how to do this because I have not done this before, but I'm going to try my best to see if we are still, able to actually find the study sheets that we were working on. I'm not sure that we're going to. We might have to, let me think about this for a second. We might have to actually get the, let me see. We might have to actually, you know what? Let's switch over from talking. We've been talking about biology. I think what we're going to do, because I just have a feeling that this is going to be easier to talk about equations. I think for the rest of this one, I think it might be easier to talk about equations. Let's see. You know what? We're going to do what we can. So there's going to be, there might be a little bit of a mismatch here on the YouTube versus TikTok side. So apologies in advance if you guys are following along on the YouTube side that this might not match exactly, but we're going to do what we can so that the TikTok side gets some questions and things answered. We'll do what we can. All right. Okay. So on the TikTok side, we'll see what we can get to display. I wonder if there's a way to zoom in. I do not know. This is interesting. Okay. Well, here we go. We're going to kind of crash through this because I know how to do this from the desktop and I don't know how to do this from the mobile, but it is what it is. All right, next thing. And guys, do ask your questions. If you have questions in the comments on TikTok or YouTube, we will still pause for questions. But for now, equations. We're looking at some of the mechanics equations. V squared equals V naught squared plus 2a delta x. This is going to be the way that you find velocity changes when you have acceleration and distance, but not time. We'll talk about that in 1 second. We do pause for questions, and we have a question that says, "What tips for coming closer to testing date?" Okay, if you're coming closer to your, let me get rid of the actual question screen here on the YouTube side, and we will talk about that right now. All right. Okay. So on the TikTok side, we have a question that says, "What tips for coming closer to testing date?" If you have, if you are getting close to your MCAT testing date, the thing I want you to keep in mind is two things. One is if you're really close, if you're less than 4 weeks away from your test, you need to have done your question bank already, and you need to be working on practice exams. Two, if you're not within, say, 5 or 6 points of your say target score, you probably need to reschedule your MCAT for a later date. I take this super seriously. I do not believe in people applying if they're more than a few points off of their goal score. Now, if you're above the 500 mark and you want to try, there's nothing wrong with that as well. All right, let me see. Noah says, why do math equations like this matter for med school? Okay, Noah, you'll have to let me know kind of what stage of schooling that you're in. But the general perception of med school as not being very mathematical is true. There's not a lot of math that you're going to need to know for med school itself. But the entrance exam, the MCAT, is definitely full of physics, and there are some of them that involve math. And the equations that you see here are for the physics section of the MCAT. All right. Next one says, in terms of approaching crunch time and how to review MCAT full lengths and keeping track of constant gaps as well as refining maybe like cardiac output. These are good questions. I'm going to give you the kind of general answer. I am going to remind you to please make sure and submit a contact form if you want to work on some more sort of specific framing it from your perspective. MCATboss.com, or drop a contact form here for help. The general answer is that if you are down to crunch time and you're doing full lengths, if you are not in awesome, I'll set you up with I'll set you up with a booking discount since you found me through the live. Just make sure you mention that you found me through the live so I remember, okay? But the general takeaway is this. If you are dealing with scores that are close to, say, the 505 and above mark, you could probably go in within a month and take it and come out with an okay score. And we can talk about strategies for timing, things like reading the problem first. How many minutes should you spend on each component of the cars section? A 507, awesome. Good job. I'm more than happy to set up a session with you. Even if you don't do a bunch of hours with me, a session or two can kind of nail down some of the time management strategies. But generally speaking, you're wanting to make sure you're not spending too much time on passages. You're wanting to make sure that you're not just reviewing for content gaps, but for actual execution, context, and habits that change your score. Please, Leah, drop me a line and set up a session with me about cars, and don't take the test with a weak car score. Please do not do that to yourself. Cars is reading comprehension. For those of you who are new to this, reading comprehension is the single most important section of the entire MCAT. If you are terrible at the sciences, you can work up to mediocre on the sciences and get a good car score and get into med school. If you are okay at the exam, but cars is not your jam, you will have a really hard time getting in. The main reason for that is that your board exams are 8 to 10 hours of reading. 8 to 10 hours of reading. And if you do not have the reading speed to do well in cars, it is a really big challenge. Is 124 too low for cars? That is borderline. I do have a student who got in this past year with a 124 in cars. They applied to over 30 schools though. So they spent thousands of dollars on apps. If that is something you're okay with, there are sometimes possibility of getting in with that score. That is borderline dangerous though. I would try at minimum to get it up to that like 126, 127 mark. But there's quite a few things that we can do to get closer to that. Drop me a line here or on MCATboss.com, and I will do my best to get back to you. I try to get back to every inquiry within less than an hour after the live. That way I can set people up with a same day discount. I really always do my best. If you don't hear back from me instantly, there may be a typo. Submit a second form or reach out to the website, okay? Mopane says, I got a 519 on the MCAT in January, but my GPA is 3.5 science. Can I go MD? Yes, you can. If you have an app that makes reasonable sense about why the GPA was at a 3.5, they're going to always have some kind of question if that is the case. If you are ever dealing with an MCAT that is not, and I'm going to switch my background. You guys can, let me see here. Just a really quick preview. I'm going to be trying to drop a cars one here really soon. Working on quite a few of these. So drop me a message if you haven't already. And let me jump through these last few questions. We're going to be wrapping up in the next few minutes. Oh, real quick, Mopane, the main thing. Oh, Mopane also says, I majored in biomedical engineering at the University of Florida. Okay, so the main thing that I want you to think about, Mopane, is you need to figure out how to explain why you got the GPA that you got. If for any reason you had a reason to get if you had a reason to get a 3.5, that is something people would understand. If it humanizes you, if it demonstrates that you have learned from it, those are all ways that you can turn a weakness into a strength, but it depends quite a bit on what you do in your application and what you do in your personal statement. For students who have high MCAT numbers, but are having a hard time getting interviews or are afraid that they're going to have a hard time getting interviews, I really emphasize get somebody experienced, whether it's myself or someone else, but get somebody experienced to help you actually brainstorm what you're going to do in order to do really, really well on your essay, and make sure that people read your app instead of dumping your app. Okay. Drop me a line if you want help reading over your essay or helping you brainstorm an essay, and I will, I'm happy to see what we can do about that. All right, guys, we're going to wrap up in the next 1 minute or so. So if you can drop any other questions, I will try to power through any other answers in about the next 1 to 2 minutes. And then other than that, I do ask you guys to head over to mkboss.com and drop me a line if I can help. Diego says, what are the bare minimum someone should have in terms of stats? Okay, so if you are looking, and we can do this the throwaway here real quick, and we'll wrap up here in just a second. But if you are looking at the MCAT GPA grid, I encourage anybody to type MCAT GPA grid into Google, and you will get the actual, don't go with whatever AI answer they give you. Go with the actual stats board of the official AAMC. And this is what you're going to find. 41.9%, 42% is the average acceptance rate for medical school applicants. And so if you want to get to that or above that, that is what I would call a good chance of getting in. So 42% means that if you are a 4.0 GPA, you're going to need in the 502 to 505 on the MCAT. If you want a similar acceptance rate and you have a 3.6 to 3.8 GPA, you are looking at a 506 to 509. If you have a 3.4 to 3.6 GPA, you are looking at needing a 510 to 513. And if you have a 3.2 to 3.4, again, you need somewhere between a 510 and a 517. And then if you have a 3.0 to 3.2, you are going to need north of a 514 to get those kinds of admission stats. All right, guys, I hope that is helpful. I'm going to leave it up for about 30 more seconds. Drop any other questions you have. Don't forget MCATboss.com. Super simple, MCATboss.com, and let me know how I can help you guys to increase your chance of getting